Good morning. Welcome to the TechGrid. I'm Tammy Schiller. I'm the director here. I'm also strategic partnerships for NOC-WD. Thank you for coming today. We have Professor Cook with us. So happy to have him here from Purdue University in Indiana all the way. He's probably enjoying our weather here. Probably doesn't recognize it. This is sunshine, sir. So again, thank you. If you need the restroom, it's straight in the back. And I'm just going to hand it over to Pat. Thank you.
For later. So it's our pleasure to have Dr. Graham Cooks. He was born in Benohi, South Africa, and he was awarded 2 PhDs in chemistry. So one wasn't enough. First one from University of Natal, and then the second at Cambridge University. He is a distinguished professor of chemistry at Purdue University, where he spent the bulk of his career. His interest involved construction of mass spectrometers and a plethora of unique applications. This has led to constructions of tandem sectors and miniature ion trap mass specs, much of which you could see on the side over here, all kind of stem in some shape or format from Professor Cook's group. He's pioneered an entire field of mass spectrometry through the development of desorption electrospray ionization, DESI, which is an ambient ionization technique. That was the first ambient source, and that's had widespread effects, even the operating room. So there are DESI mass spectrometers for cancer diagnostics, brain surgery, you name it. More recently, and much of what his talk today is going to be about, will be the discovery that microdroplets and confined volume reactors produce products at accelerated chemical rates. Dr. Cooks's lab is named Aston Labs after Francis Aston, one of the grandfathers of mass spec. So every student who has gone through his lab, visiting scholar, postdoc, visiting faculty, receives an Aston number. And I think this is pretty impressive. His most recent student, I think we were just talking about it, is 388. So 388 students, postdocs, visiting scholars have come through. And out of that 388, 187 of those are PhD students. So you could imagine the widespread effect of that. My own Aston number is 320. So going along with this, Dr. Cooks has published over 1,700 manuscripts, is one of the 100 most cited chemists in the world. His work's been cited over 100,000 times. Again, I could begin to list all the awards, but you'd never hear his talk. So just a few: the Othmar Gold Medal by the Science History Institute, the IUPAC Analytical Chemistry Medal for Lifetime Achievements in Research, the Dreyfus Prize in Chemical Science for groundbreaking work in mass spectrometry. He's a member of the National Academy of Sciences, a fellow of the National Academy of Inventors, and a fellow of the American Academy of Arts and Sciences. We are very much looking forward to your talk, sir.
So I'm very, very pleased to be here. I'll just start with one small correction. The lab was named after not Francis Aston, the Nobel Prize winning physicist. It was named after a pet hamster that we had that we called Aston. We had various pets in the lab, mostly because you weren't allowed to have pets in the lab, so we did that. So we also had a cat called Fred McClafferty and a dog by the name of John Bynum, and a variety of other fauna and flora. We had all sorts of flora in the lab, including something like 400 people that have just been named. Some of them were really, really smart. Some of them were not really, really smart, but they were all really, really nice. And when you've been associated with science for as long as I have, that is the quality that counts as number one. The other things are very desirable, people being able to work really hard, come up with new ideas, new instruments, new experiments. I want to say a very, very heartfelt thank you to this group, to this laboratory. I've been treated very well in a very short period of time. So thank you to the leadership. Thank you for the warm welcome. And I appreciated very much the short opportunity that I've had to interact with people here, and want to convey some of my feelings about the subject of science in general, but also particularly with regard to mass spectrometry and analytical chemistry. So I'm talking from a vantage point of somebody who's done mass spectrometry for 60 years. Mass spectrometry is only 120 years old. And so I've been involved in mass spectrometry for 57% of the entire length of the discipline. And so in that period of time, I think I've learned a little bit about the subject, and been quite amazed at the way in which it has grown, not steadily, but actually in spurts. So something happens, in particular, there's a new, either a concept or an instrument, and the concepts have to be quite targeted concepts, and then there's a spurt. And so after a spurt, of course, there's a plateau of some sort, and then another. So maybe I should talk about when there weren't spurts, when there were plateaus in the 80s, before the development of electrospray, which everybody in science, I think, knows about, the electrospray methodologies, which produced proteomics, metabolomics, and this huge revolution in biology. Before that, we were in a situation in mass spec where, yes, people were looking at larger and larger molecules, and the resolution was going up, but it was a bit like the car manufacturing industry. You know, year after year, not much happened. They changed the shape of the grille. We changed the performance spec slightly and so on. And then the development by John Fenn of electrospray ionization changed everything, because now you can ionize and use mass spec to study any molecule that you wish. And so now we're in the mega Dalton range, 10 megadalton, tennis balls, golf balls, whatever you want to put into your mass spectrometer. In principle now possible, certainly individual blood cells, for example. You can mass analyze that complex material and obtain some information from that. So what has mass spectrometry given us? Briefly, this is, I said, from the point of view of 57% of this time, fundamental physics and chemistry. So isotopes, things we won't even think about on a daily basis because they're so deeply embedded in science. Thermochemistry, for example, very strongly dependent thermochemical values on mass spec. So that was the first era. The second era was this, what a lot of people think about mass spectrometry as it is now, as analysis. And initially this was forced along really by the environmental problems. There was a huge push from environmental analysis, organic structural analysis, but more recently the omics. And I'd take that as being the present, but not the future. And then the materials sciences. So 1 in 10 papers published in the journal Science or Nature uses as an essential part of the data that goes into it, mass spectrometry data. It's not usually labeled. There's not a special section to say, hey, we're doing mass spec, but they surely are. And this is isotope-based determinations, whether it's of age or some kind of a biological or a geological process. So the effects are deeply felt in material surface and isotopic science areas. And then looking towards the future, I think that synthesis, organic synthesis, material synthesis will be an essential major component of mass spectrometry. And so that's a prediction, an expectation. And that's because mass spectrometry has never been about really only measuring masses. It's a manipulation. It's a study of the chemistry, the physics of ionized materials. We have a little argument with the electrochemists about that, but we'll stick by our guns and claim the subject of ion chemistry is mass spectrometry. And so for that reason, the fact that we can use ions made by mass spec methods and do synthesis, green synthesis, without catalysts in droplets has got to be of some significance. And that's an important part of what I want to talk about. So the history of mass spectrometry to some degree is the history of how do you make ions in the first place. So this is a very busy slide, so don't look at it except maybe to notice a photograph of Nobel laureate John Fenn, who invented this electrospray method. And the electrospray method is a soft ionization method. Matrix assisted laser desorption was another. That's a generation ago, those soft ionization methods. So where we are now is with ambient ionization techniques. So what do we mean by ambient ionization? We mean ionization methods where you don't have to process the sample. So if we go on and talk about one of those ambient ionization methods that we call DESI for short, we have, for example, some biological botanical samples and we want to study them. We can, of course, dissolve them up and then electrospray them and do the standard ESI electrospray experiment. But it would be much more straightforward if instead of that, we could do this experiment shown here on this cover of Science magazine, of simply spraying the object itself. And so ambient ionization is recording mass spectra, creating ions from the intact objects in their native environment. So, and obviously there's huge implications of that to any object in any environment, and using the power of mass spectrometry to do chemical characterization under those circumstances. So what you're seeing on this slide is the fact that not only can you, not only can you spray an object, that you can spray in detail at various positions on that object. So for example, you can take a fingerprint, a virtual fingerprint, one that I've just placed on this wooden table here, and spray across that fingerprint with a finely focused spray of droplets. We're using droplets, not photons, to spray and to record information, chemical information, pixel by pixel across that fingerprint. And so by moving a sprayer like here across this fingerprint, back and forth, you've got very sensitive computer here. You get a mass spectrum at every position, and you convert that mass spectrum at every position into an image, and that image can be an image of all the lipids or a particular lipid and so on. So the experiment is shown best perhaps in the simulation that you see at the bottom, where we've got a droplet solvent hits, splashes. So what is this experiment? It's microextraction. It's simply the extraction of chemicals from a position by a solvent. Nothing more than that. Why is it important? Well, you can extract chemicals from virtually anything with an appropriate solvent. You can do it as a function of position. So now you're doing molecular imaging, which is a tremendously important part of biology at the moment. But at the same time, you're getting this detailed chemical information on an object, on a process. So the experiment may be looked at slightly differently. Here's a version of the experiment. What we're going to do is we're going to take a compound or a collection of compounds as shown here, and we're going to direct solvent at a particular spot. We'll see, we'll have those secondary droplets generated, and we can take those droplets into a mass spectrometer, and that's a chemical analysis experiment. We get information on what was there. We can also, however, collect those droplets. If we collect them after they've traveled a very short distance, we will collect the species which are on the original surface. However, if I increase that distance so that the droplets have got time to break up, and to form smaller droplets, which is a standard process in electrospray, then there may be reactions between those components. And what I land on the next surface may actually be the products of reactions. So this is the most critical point in this talk. Mass spec is about analysis. Yeah, but not always. You can do analysis if you like. You can spray on a surface, splash, extract, and measure. Maybe nothing happened in that process except you dissolved. You dissolved the compounds that were on the surface and you measured them. So what? Well, you got a good analysis. It's biologically interesting, maybe medically important, as Pat just mentioned in the introduction. But what maybe I want to do is to synthesize new compounds. If I want to synthesize new compounds, how do I do it? I get one of the hundreds and hundreds of round bottom flasks that I saw in the storeroom back there. I mix A and B, and I take a little time, nothing happens. Okay, I add a catalyst, add a little bit of heat, take a little bit more time. And after 3 hours, I can do an extraction, separation, and take a bit more time and get an NMR spectrum and characterize the compound. An alternative to that may be to work on a micro scale, a very small scale. That's what we're aiming at to do here. So maybe I can do this experiment in which on the surface, I've got A and B. I send the droplet of solvent in, extract, and now the secondary droplet contains A and B. And if it's a small droplet, so there's a lot of surface area, and for reasons that I'll explain in a lot of detail, the reactions are accelerated, and A and B that don't react at all in a week standing in a flask react in a millisecond as they travel from that surface to a collector or whatever else you want to do with them. This is, so if there's any question about what I've just said, this is the time. Now is the time. And I would really like you to interrupt and ask questions now. Two types of mass spectrometry: analysis, synthesis. Same instrument, same conditions. The only thing I've changed is I've changed the distance those droplets fly. And I change the distance, I can actually follow the course of a chemical reaction just by measuring as a function of distance. One of the problems with talks nowadays is that it's all about slides, and I don't want to talk in a way which is not all about slides, because you get blinded by slides, right? And so I could show you lots and lots of, I'll do that later, show you lots of things that you don't want to see. But for the moment, I'd rather talk to you. Yes, thank you. Yes, no, that next slide, one afterwards, it's coming. But yes, it's limited by that, but it's a small limitation. Yeah. Yes, thank you.
The two starting materials, the distance between the droplet to the mass spec? Like,
yes, the two starting materials are already mixed on the surface that we're impacting, as you see in the current slide. The distance that those secondary droplets, which you saw in the previous slide, travel before they are intercepted or collected. That's the distance. It's typically, if I want to do ordinary analytical boring mass spec, I do 2 millimeters and I just go away. And so, yeah, you can have it. It's being sold by 6 manufacturers and it's everyday item of commerce, $3 billion a year market. It's boring. So if I want to do something different, I try to do synthesis, which means I've got to stretch. And I stretch from 2 millimeters sometimes to 50 centimeters. If I stretch that far, of course, the droplets are, they highly dilute by now and they spread out and so, but yes, exactly. Okay. Yeah, there's another question. It's much better to have questions than to give a slide talk.
So you mentioned having your A and B
Yeah.
on the plate already. would
Yeah.
there be a situation where you'd have A on the plate and you'd be spraying B?
2 slides coming.
Okay.
This is great. Great. You can do it both ways. There's a question here.
I was wondering, have you ever tried incorporating like a catalyst somehow into the process, either in the well plate or in the
Yes.
mixture?
We have. And the reason for not doing it is that people are easily confused. And the scientific audience in particular is really easy to confuse. And what they then conclude is that microdroplets depend on catalysts. And to make a point that it is not, they do not depend on catalysts, that we've got tons of reactions occurring without catalysts. I refuse to have any catalyst anywhere near any of our experiments. It's a psychological reason, not a scientific reason. Yes.
Where in the droplets is the reaction occurring?
The reaction is on the surface of the droplets, and you can show that by systematically changing the size of the droplets. Most of these experiments don't have control of the set of droplets. If you do single droplet experiments, you can have control. And that can show you that as the droplets get smaller and smaller, the reaction rate increases. So droplet size experiments demonstrate that a big droplet has very little surface. A big droplet would be, for example, 5 millimeters size droplet, with a huge droplet, no acceleration to speak of. The bulk of the droplet is material in the bulk is reacting at bulk rates, but the difference between the rate constant in the bulk and the rate constant where for the surface can be 1,000,000 times. So if you find something which is a factor of 1,000,000, you should get very excited. And that happened in 2011 when we first encountered this and published it. But that paper doesn't get cited. It's what happened subsequently once a group of people joined this enterprise and so on. So yeah, great questions. Yes, thank you. Yeah. Right. Yeah, because it's quite difficult. I'll just answer briefly and we can come back to it later. Field ionization is difficult to implement. That's why, and it was one of the methods, I had it on the previous slide, a key method. In fact, the first method that was applicable to involatile compounds was field ionization, 68 Becker. Yeah, so that's the main reason. So there's a wonderful, so if we ask this number of questions for each slide, it's probably Tuesday of next week. Okay, so we mentioned this thing about applications of this method in a variety of places, and we've done some stuff with regard to brain cancer in operating rooms. I'm not going to go over it in any detail except to show you that there's a little mass spectrometer there. It's being used to perform DESI. And then in the right-hand corner, you see a couple of spectra, a blue spectrum and a red spectrum, the one corresponding to healthy tissue and the other corresponding to diseased tissue associated with an oncometabolite associated with a change in the gene. And they're very, very easily distinguished. There's a factor of 50 in the intensity ratio of those two sets of peaks. You get this kind of a situation very rarely. But this is all the other characteristics of this DESI method show up here. It's very simple to implement, very fast, and quite precise information. Okay, so you're up for this kind of general discussion that we're having. So I will ask you about the origin of life. What are the essential chemical steps? So this is a diversion. We already had a lot of diversions. Essential steps in the origin of life. Well, okay, you've got to make, for example, proteins, which means you've got to make peptides. How do you make peptides? From small molecules. What small molecules? Amino acids. A common idea is that this chemistry originated in water, seawater, droplets of seawater. So you had amino acids. Yes, you still do in the same medium. So what are you trying to do? Well, you're trying to form a peptide bond from two amino acids. Amino acids forming peptides means you're removing water. You're trying to remove water in a water solution. Okay, so that thermodynamically doesn't make any much sense. So it's a paradox according to Veronica Weider, thermodynamically prohibited. So how do you get over that paradox? Well, you conclude, in fact, that the droplets of water are not actually wet, they're dry. What? They're dry on the surface, which is where we said the reactions are taking place. So if they're dry on the surface, it means that there's not a high density of water per se. So what there are, in fact, are hydronium ions isolated and a couple of hydroxide and so on. But it's better to say water is dry because I get your attention. Okay, so they're not, I can say it this way, they're not wet everywhere. They're wet in the middle, but they're dry on the surface where the reactions are taking place. So let's try an experiment. So this is data from 2 days ago. Here's an experiment in which we're taking carboxylic acid, which happens to be an amino acid, and an amino compound, an amine, which happens also to be an amino acid. And we're trying to remove water from them and doing it in ACN or in aqueous. In this particular case, I'm going to show you in ACN. So we did that experiment and we get a product which corresponds in mass to the product in a particular case of water loss. And then here's the data which makes the point that makes the point that I was trying to make before. The top figure, called 3 millimeters, shows what happens when you have that distance that we talked about at 3 millimeters. Well, it's not interesting what happens. You see a peak for the carboxylic acid, 166, and you see another peak, which is sodium attachment to the carboxylic acid. So the only ions in that spectrum are the amino acid itself. Then you increase the distance to 2 centimeters, and now you begin to see a little bit of something at 221. You increase to 6 centimeters, this thing at 221, which is the product, the amide. So there you've got your amide. So you can, and what happened in those 6 centimeters? You had some droplets, and they've got charges on them because naturally the droplets will have surface charges, and they started to coulombically explode. They subdivided, and so you get large surface areas. Okay, so I thought there might be students here, so I tried to sympathize with them. So chemistry is so complicated, isn't it a pity, and so on. But this chemistry that I want to talk about is not so complicated. And so I'll give you 7 reasons why it's not complicated on the next slide. Thinking about Shakespeare and the 7 stages of man, including people that have been 55 years in mass spec. So this is a summary. Microdroplet reactions occurring at the interface are fast. There's an acceleration factor. They're mild. We often do not have to do workup of the products in the usual way to get a good spectrum, a good NMR spectrum. They're green, no catalyst, we talked about them. You can easily scale them up. You can scale across. You can create large numbers of reactions. You make it do an array. You can work at very low amounts and you can look at a wide range of compounds. Okay, so we have to speed it up because you're not listening fast enough. This is the history of mass spec coming beyond the ionization methods that we talked about before and we're now in the era of DESI. So DESI was developed as an ionization method, first published in 2004. It was used for the next 8 or 9 years to do derivatization reactions. So these were accelerated reactions, but not recognized as being, the acceleration wasn't explicitly recognized. Most of that work was done in my group. And after the first publication on accelerated reactions in DESI 2011, I've got this purple bar because that's when a lot of this chemistry was worked out in detail. This is that first study that I mentioned. So now we're bringing in, this question about, can we bring in one reagent in the solvent? So we're spraying with A and we've got B on the surface, and we're making C because C is present now as a result of A and B being in those tiny little secondary droplets that are being pointed to in that figure. So this experiment was the first accelerated reaction experiment. And here's the second, 3 orders of magnitude, 1,000 times. And here's the second one. This is no longer DESI, this is electrospray. And so in this experiment, the spray was done with a mixture of the 2 compounds that are shown, a ketone and an aldehyde. This is called the Kleisen-Schmidt condensation. It's another condensation reaction, loss of order. And the illustration there is meant to show that the droplets are getting smaller and smaller. Stop doing that. The droplets are getting smaller and smaller, and we're getting product in quite large yield. So what I conclude from this is that this may represent the next revolution in chemistry. And it came about from a combination of 2 things. The first is an ionization method in mass spec used to create these droplets that are going to be the reacting species. That's DESI. Secondly, the fact of there being a phenomenon which hadn't been realized before, which is this acceleration at a droplet interface. And in fact, contain water, because water is the basis for the reacting species in these experiments. That combination then leads to high throughput analysis, synthesis, and bioassays. So I now want to go into this possibility of doing large numbers of reactions by these methodologies. The kinds of reactions are sort of illustrated here. So the 10 to the 5, 10 to the 4, 10 to the, and so on. These are the acceleration factors. So the smallest is 10 to the 4, the largest is 10 to the 6. So these are the rate acceleration factors by which a variety of ordinary organic chemical reactions can be run. So the main audience for what I'm saying is not analytical chemists at all. It's synthetic organic chemists who should be paying the most attention to this, because it's doing what they do all the time, except in a new way, as we've said, without catalysts, with product relatively pure and very, very fast. Here's the scale of the experiments, just to make sure that it's clear, all the way from nanograms up to, in this case, 50 milligrams per minute. And Pat has gone a whole lot further than that in terms of grams in unit time in order to make these species. It's not restricted to organic these advantages are not restricted to organic reactions. So I'm going to show you a very different example, nanomaterial synthesis. So in this experiment, which is again extremely simple, chemistry is very complicated, not always. This is as simple as you can get. Take a nanopipette, put an electrode in it. That electrode can just be an inert electrode, but it could also be made of the material that you want to study. Or else you can put a solution of a silver salt. So you can have a silver electrode, or you can have a silver salt with an electrode in it and spray. You'll get droplets. What will those droplets contain? Well, those droplets will have silver ions in them. Now, some of them will be positively charged. Some of the droplets will be overall negatively charged. They'll have an excess of chloride. You've got these silver pluses and chloride negatives. So if we go ahead and arrange that we're only going to look at the positively charged ions, then we've got a bunch of silver ions in solution. When they reach the surface, they are discharged. They become silver. Those silver atoms will associate. So this study in 2016 was the second study that had ever shown this phenomenon. The first one we published a year before this, making nanoclusters, nanomaterials, simply by spraying from a solution, nothing else. And if you look in detail, and that's why I chose this one, this is on the micron scale and this is on the nanometer scale. So what are these silver things? They're pearls. They're 60 nanometers or so, 6 nanometers or so, pearls. So if you want to give somebody a necklace, so this is my donation to that. You can have as many as you like. They're all about the same size, like pearls ordinarily are, and they're pure silver pearls. And they're made by just turning a switch. There's nothing complicated. This is a high school, it's a pre-high school experiment. Let me show you some context then for this experiment. So what I just showed you was that experiment in 2015, published in 2016, and the year before, the synthesis of gold nanoparticles the same way. So it's a general process. What do we learn from that? We learn that if we want to do something really different, why don't we go ahead and do it? So so now I turn it over to you. I've just showed you can make nanoparticles this way. What nanoparticle material would you like to make? Yes. Oh, wow. Yes, I don't know how to do that. Anybody else? It was so good the last time. What are the strategic metals that we worry about? Aluminum. Did you say aluminum? I didn't know it was. Is it a strategic metal? Yeah, but it's not in short supply or local. No, yeah. So we worry about the ones that we can't get. So what are they? Yeah. The rare earths, right? The rare earth metals. So we should do this experiment on rare earths. And I don't have anything to show you, but I do have lithium to show you. So we do this experiment on lithium. So we deposit lithium metals from lithium chloride. There's a lot of lithium chloride. We've got a shortage. We're digging up half of Nevada to get lithium. So why don't we just take a bit of ocean and do this experiment? So this is the result. So you can look perhaps at the overall process. It's done with nitrogen because we were actually trying to make ammonia. And so in this experiment, it's the conversion of nitrogen to ammonia, which is a perfectly good microdroplet experiment. But it was done in the presence of some polymer, excuse me, in the presence of a polymer. So we did in fact form lithium metal, but protected it because it's in water. So this electron energy loss spectrum shows that you've got lithium metal hidden in the polymer that was present in this experiment where nitrogen was being converted to ammonia. There was too many stories at once. I want to take a moment to very quickly show you what I think about the mechanism of this accelerated reaction. So there are key intermediates that are needed. They serve as the catalyst, but they come from the water in the droplet. That's the first step. And then there's an acceleration of rate, which is the result of partial solvation, and I'll make that clear. So the primary species that I believe are involved in this chemistry and all this chemistry are water, iron, which is shown at the top, and the electron formed by the ionization of water. And in fact, since we had a question about field ionization a moment ago, formed by field ionization of water by the electric field, which is widely believed to exist at the surface of microdroplets. So the electric field is formed spontaneously, positively charged ions in a droplet which has an excess of positive will go coulombically, repel each other and go to the surface. And then there'll be a counter layer of negatively charged that sets up an electric field, which is quite strong and could be involved in field ionization of water to produce those two starting. The next important step is under item 3, that water radical cation solvates and generates a hydronium ion, as you can see, and a hydroxy radical. And the hydroxy radical is an oxidizing species that gives rise then to hydrogen peroxide. So Dick's there, who's going to be talking in this group in a few months' time. We'll talk a lot about oxidation and reduction reactions in microdroplets. And this is the suggested mechanism that we've published on this. The second step, the actual increase of reaction rate, comes from a 2016 paper by two of Pat's compatriots at Purdue, Yan and Bain. And the suggestion in this paper is that because these molecules are not solvated in the usual way, they're actually accessible to each other. Reaction rates should go up. It's a very, very simple qualitative argument that hadn't been made before. The rates should go up, and in fact they do. And there's a bit of thermochemistry shown in top right of that figure. And then this is the field ionization mechanism, just saying what I just said. You've got these microdroplets, you've got the strong electric field, you generate water radical cation and electrons, and you're on your way to doing the kind of chemistry that led to all of those reactions, including amide bond formation that we showed early on. So we were lucky enough in the period 2016 onwards to be funded by DARPA. When did we start? 15. Can you double that? No. No, don't worry. To be funded by DARPA for high throughput, a program called Make It. And DARPA is a phenomenal agency because they have all these ridiculous ideas, many of which have led to very, very useful things, as everybody knows. And so Make It was make these pharmaceutical agents in the field with these simple ingredients. And we had no idea how to make anything at that time. But we'd just come through this period I've just described where we saw that these accelerated reactions were occurring in droplets. So we built a system which took a large number of separate reactions, mixtures, and did what I've showed you already. So this rest of the talk is all boring because it's straightforward application of everything I've told you. So if you've got a plate which has got 6,144 reaction mixtures, which you can make automatically, which we did, standard size that we use, then you can spray each of those in turn. And if you take half a second for each, you'll do 6,000 experiments in an hour, and you record all that data. So this system does that. So now you can explore a lot of chemistry, because you can take, if you're clever enough and you know 6,144 name reactions, if you've memorized them, then you can do 6,144 experiments. Or if you know one name reaction, you can do 6,144 replicates. And you can get high precision in whatever measurement you're making. Or if you're just an average person and you know 20 reactions and you know 36 replicates is a good number, then 20 multiplied by 36 is not 6,144, but it's close. Okay, so that system was followed by this system, which we're working on using currently. Nicholas Morado is the postdoc assistant research scientist who's been most responsible for this system. And you'll see a few things that perhaps you recognize. And here's a plate, which is a piece of glass with Teflon on it. And it's got, in this instance, I think in that there may be 6,144 spots, individual spots. They're 1 millimeter apart from each other. Each spot has got 5 to 10 nanograms of material. And we're going to do quantitative chemistry and biochemistry on those spots by spraying, the way we've been talking about it. And that requires a bunch of equipment that's shown there. But ultimately, we're really just doing the same thing I've already talked about. We're creating droplets which contain, firstly, the reagents, and as they shrink and explode and have more surface area, these reactions go faster and faster, and we end up with products which we can collect and characterize as indicated. That means we can do this kind of super interesting chemistry where we take a bunch of reagents and mix them with biological compounds, drugs. And here we took 22 drugs and sought to modify those drugs, which means making new drugs with a bunch of reagents, undergo 20 different reaction types. And for each reagent, we used a whole bunch of different. So if we're going to convert ketones to imines, we want to use a whole lot of amines to make the imines. So we did the chemistry that way. So that's the reason for the scale being like this. And this is the kind of chemistry that we did. So I'll point out then that the success rate on the 5 or 10 nanogram scale here is about 50%. So our choices were pretty good. Half of the reactions that could have happened did happen, and we got enough material to be able to characterize the products. And in favorable cases, we got enough material to go on and do bioassays. So analysis, synthesis, and bioassay, it's the triumvirate of this kind of methodology, this kind of instrumentation. And Pat, of course, has got an instrument of the same type, which does the same things. You can do the analysis, you can do the synthesis, and then you can do the bioassay, which really tells you if the new compound has got any activity that's worth pursuing. You can do all of those things very fast. Then you can decide whether you're going to go ahead and synthesize the compounds that you need by standard routes and do standard testing. So it's all about the speed of reaction. So the next few slides are only for the people that are at all interested in bioassays. You can rest your eyes for a while. So on this slide, we try to modify antibiotics. Who wouldn't want to find new antibiotics? And the experiment involved modifying and converting amines to quartz, converting amines to pyridiniums, and so on. You can read these faster than I can. So there's 7 or 8 different, 11 antibiotics, 55% success rate, 1 second per reaction. And the heat maps indicate whether a particular amine, if it was an amine, a particular nucleophile, if it was a nucleophilic substitution, whether they reacted or not within the constraints of what we're talking about. Then we had a visitor came in, was interested in antifungal agents. So we got a standard antifungal compound. And we said, okay, we want to impress this person, comes from a company who would really like to work with them because we think we can help them. And in 90 minutes, how many new, starting from 0, how many new antifungals can we create? And this is however many. So we did 167 reactions while the person was waiting. And some of them were quite easy. Of course, for example, this antifungal, amphotericin B, has got a lot of diols. And so we converted those into boronic acid esters for the organic chemists, or if you can stand it, or else into various other compounds. So here are the people that did that experiment, and the success rate is very similar. So we could make these compounds. Are they good antifungal agents? What Alexis and Kittman did with the antibiotics is they did standard antibiotic assays when we made these compounds. Almost all cases showed very much the same antibiotic activity as the known compound. A few cases showed much less. We direct the activity. And a couple of cases that are still being studied showed what appeared to be enhanced activity. But the antibiotic testing that we did was standard antibiotic testing. We can also do ultra-fast antibiotic testing by methods that I'm not going to tell you the story of adipic acid, 6-electron oxidation. I'm going to skip that one. I'm going to mention to you that in organic synthesis, if you can't show an NMR, you haven't shown anything. So the fact that we can do mass spectra and demonstrate by MSMS that we've got certain compounds doesn't count. So we have to scale up in order to talk to the synthetic. So here's the scale up of the adipic acid and the NMR. So how long does it take to do the scale up? It takes about 10 minutes, 5 to 10 minutes to get this kind of quality NMR, but no workup. So you do the reaction just by spraying, or else if you use multiple sprayers, divide the amount of time by the number of sprayers you use. If you use 128 sprayers, then you take a minute. That's Manish's work. I'm going to skip. This is all more synthetic organic chemistry, green synthetic chemistry. If there's anybody listening outside, then maybe they're very interested. Nitrogen coupling, Buchwald chemistry. Or maybe I should just point out that we're trying to diversify when we're coupling, and we're removing halogens and replacing them by nitrogens. And here's the sort of success rate that we're having in these experiments, widely successful. I'm going to skip that. I'm going to skip that. I'm going to skip that. Okay, so we've got 3 more slides to talk about. This is the end, almost. So the bioanalysis, so I've emphasized the third leg of these experiments is to do bioassay. And a lot of this really comes down to doing enzyme kinetics. And we run a facility based on the methodology, based on this instrumentation. And most of the requests relate to enzyme kinetics, Michaelis-Menten characterization, which is what's shown here, plotting inhibition or reactivation of an enzyme using an appropriate drug or drug candidate. This is the basic work. And I claim for the quality of this work that we're doing something which is really quite good, using multiple samples, 1,500 samples or so, 20 minutes, and getting high quality Michaelis-Menten kinetics from different species. So this is intrinsically interesting. It became even more interesting when we looked at Series A chemical warfare agents, where the question of binding and release becomes paramount. So you've got agents, the A series drugs, and the appropriate corresponding agricultural phosphorus-based agrochemicals, which will do the same thing, binding to acetylcholine esterase. So using the second system that I talked about, which is based on ultra high throughput, much better than one sample per second, DESI reaction, we can look at this problem, the problem that acetylcholine esterase will be inhibited by an A series agent. It may go on and undergo an aging process, but what we really are interested in is medical countermeasures, release agents. And at Purdue, we don't work with Novichok, which is the A series agent that was being used in this work. We have a collaboration with Lawrence Livermore, National Labs, people mentioned in the reference below. And that led to the combination of our experiments, this ultra high throughput DESI on the bound species and their work, not quite as fast at the time, maybe faster in the future, on the agent itself, allowing us to measure the binding, the release, and the activity, and the degree to which different countermeasures would be successful. So this work was published last year in PNAS, and the task at hand right now, obviously, as in any drug discovery project, is to improve the quality and the effectiveness of the medical measures, the drugs, or in this case, medical countermeasures. And so a whole series of experiments, some of which are looked at in this data here, are involved in reactivation of the enzyme using modified versions of species which are now commonly used, the oximes, which release the phosphorus binding molecule. So I mentioned that we have a core facility, and you guys have your own facility here, so I'm not advertising for you, but maybe Pat doesn't mind having more requests for people bring him samples to work on, especially this bioassay side, which I don't think is done a lot of. So the full spectrum of making new compounds by new methods and then bioassaying those compounds. And I'm going to leave it there to just simply summarize by saying that this is not really exclusively in any way about mass spec. In many ways, it's really about new ways of doing chemistry, new ways of doing synthesis. And it's wide open, as I hope I've shown, in terms of the kinds of compounds you can make, the amounts of those compounds you can make, and the speed with which you can interrogate the activity as well as the synthesis of compounds. Thank you very much.
If you've used salts for salting out your analyte of interest within the droplet.
One of the most interesting features of DESI is that it's the only mass spec method you can use with ordinary biological buffers, phosphate buffers. So we often use salts in amounts that are huge and would immediately stop an electrospray mass spec experiment. And this is because DESI doesn't have the only thing we spray is pure solvent in the usual experiment. So nothing can get blocked. And so that's the usual problem with salts.
Hi, Graham. Thank you for a great talk. I have two questions for you. My first one, you mentioned how water plays a role in the ion chemistry in the droplets, but most solvents used in electrospray don't have a high content of water.
Can I answer that first?
Oh yeah, yeah, go for it.
We went mostly in the period 2011 to 2015. We were using organic solvents exclusively, but we were in a hurry, so we didn't dry them rigorously. When we started to dry them rigorously, all the reactivity went west. Okay, from the east, it went west, came to you. So you have to have water, and that's subsequently been shown clearly. Dick Zeh, when he started doing these experiments, started with water. And so he always had water in his experiments.
And so the trace amount and
Trace
the
amounts
wet
all you
solvent
need.
is okay.
Interfacial chemistry. That's all you need. Your second question.
The second question, I'm still trying to think of how I'm gonna say it, but I'm wondering about the other factors that are in the droplet that might contribute to the acceleration. I don't know a whole lot about the field, but in the droplet it said the pH can change, especially if you're changing the distance, desolvation, charge buildup on the surface. But you believe that it's the gradient that pulls your ions to the surface, or
I think that electric field explanation is widely accepted. That is the major factor. The pH has minor effects in most reactions. The pH of the bulk, because the reaction's not occurring in the bulk. If you're going to do positively charged ion chemistry, even a trace, the trace that comes from the autoionization of water is enough to get you a whole lot of protons, which are highly, highly acidic. We measured the pH in that 2011 experiment. We measured the pH with paper, and it was 0. It was way super acid. So you got super acid. What's the point of adding acid? You don't need it.
Okay, thank you.
Mechanistically, why do unimolecular reactions not enjoy the same enhancement as bimolecular? Because they too depend on charge accrual and dispensation.
Yeah, that is explained in that little summary that I had in the corner of that slide. Unimolecular reactions, it is because of the extension of solvation over the transition state, which includes both reagents, that drops that energy. This is the factor that gives rise, and that term is in the exponent of the rate expression.
If there's an effect from the gas that's surrounding the droplet, like if you do these reactions in air versus an inert gas, is there an effect on the reaction rate?
I don't know if that's clearly enough. It's clear enough. Certainly, if you've got oxygen there, in some reactions, the oxygen gets involved because it's highly reactive. So you've got air, you've got oxygen, and so on. If you exclude, if you leave nitrogen there, nitrogen is also reactive under these conditions. We've got a species, the water radical cation, which is highly reactive. nitrogen can pick up electrons. It can also do a charge exchange. So if you have two liquids, you've now changed the situation. So the on-water chemistry, which in the historical slide I pointed out, is on water has been known for a long time. There are small acceleration factors between two liquid solutions, immiscible solvents.
Is there a parallel drive to exploit droplets as photochemical lenses and as photochemical filters using solvents that can filter out various wavelengths?
I don't know of that. I don't know of that occurring. So I don't think so.
Can you control the electrical field, or are you just aware that it's present?
No. The understanding, Wayman is the person that's done most on those electric fields. He's got several really nice papers on it. The understanding of the situation, of course, there has for a very long time been second harmonic generation studies of liquid surfaces. It's not the same, though, as a surface which is highly curved. So SHG studies on micro droplets haven't taken place. And so we don't really know in detail how to control that. No, I don't know. I think it's reasonable to propose that there is order, tiny clusters, tiny subclusters, that there are hydronium, hydroxide, hydroxy radicals, all in that space.
The person you mentioned who invented mass spec, how did he come about it and where was he located?
Person I mentioned was Aston, and he was located in Cambridge. And he built arguably the first mass spectrometer, although there were other people in parallel. An argument could be made that mass spectrometry was invented by a guy called Wien, W. Wien, in the 1880s. The Wien filter is named for him. And very much like it happens in almost all cases, a field is not often invented by one person, maybe Einstein accepted. There's some activity, and the activity, it benefits from the communication. And so the study of electrical discharges in magnetic and electric fields, and in the physics community in the 80s and the 90s, the previous 80s and 90s, was alive. The person who made the largest contribution as recognized in the English-speaking world was J.J. Thomson, who's known for the invention of the electron, discovery of the electron. More significantly for mass spectrometrists, he discovered the mass spectrometer. Aston was J.J. Thomson's student. So we need to give primacy to the professor, not to the student. After all, the student had a rodent named after him. Professor didn't. So, inside joke.
Can't hear you.
So I think that's the answer. I appreciate the historical question.
Oh, thank you.
Yeah.
So much. What a wonderful talk. And thank you to our audience for having such great questions. I can always count on you guys for giving our speakers such great questions. And I think that that's really important for them. Thank you again, Professor Cooks, for coming all this way. We really appreciate you making the trip out here. And I know that Pat has a great tour for you set up. And so really appreciate you making the trip. And I hope you enjoyed the rest of your tour. For our folks in the audience and online, our next Distinguished Lecturer Series will be March 30th. We're having Professor Adam Matzker from the University of Michigan, and he will be speaking on Outside the Blast Radius, Explosives, Discovery, Unburdened by Knowledge. So I want to see all of our research department as well as our energetics department out here. Let's fill this place up again. So spread the word, distribute this information widely to your friends. We will again get it out, mass distribution as far as we can, but please tell everyone. So this one is a different topic than we normally have, so we're looking forward to it. Thanks again. Please, Professor Cooks is going to be hanging around, so if you have some questions that you didn't get to answer during the talk here, I'm sure he's going to be hanging around for a little while longer. So thanks again, everybody. Have a good rest of your day.